going to continue on in Chapter 13, looking now at the mechanism of DNA replication. So we're going to zoom into one of those replication bubbles and see what's happening there, including several different enzymes and molecules that all together help out with this process of DNA replication. Now, as we just discussed in the previous lesson, all organisms have to be able to replicate their DNA, including prokaryotes and eukaryotes, but there are some differences in those processes between prokaryotes and eukaryotes. And so in order to simplify this, because it's already a very complex idea, we will be modeling this for a prokaryote. So just know that in addition to what we're learning here for prokaryotes, there would be actually more involved for a eukaryote, but we won't be going into those details. So let's start with the key players that are involved in DNA replication. On this diagram are several enzymes that we're going to learn about today as we step through the process. And I want to start here with the ones that are involved in the very start of DNA replication, when the DNA double helix is still one double helical molecule with all of the parental strands connected using Chargaff's rule. Remember, A pairs with T, G pairs with C. So you can see that over on the far right. That would be like the parental DNA double helix there, still together and still wound up into that helical pattern. But if we now focus on how that becomes unraveled and unzipped like a zipper, there are three players involved there. So one of those enzymes is helicase, and that's shown here as this blue triangle guy kind of wedged in. So helicase's job is to untwist the double helix and to separate the DNA strands at the replication fork. It's helping open up that replication bubble. But to prevent those bases from collapsing back on themselves and remaining open for DNA replication, that's where the single strand binding proteins come in. They bind to and stabilize the single strands of DNA so that they can each be used as a template for replication. And then while this is happening, topoisomerase is another enzyme that prevents the DNA as it's unraveling and untwisting from over twisting upstream of this replication bubble. So if you kind of think of what it's like to unravel, I don't know, rope that has like a bunch of knots in it, or if you're decorating for a holiday, you get the lights out and it's from last year and you thought you wound it up, but as you're unwinding it, you're actually creating more kinks downstream, more knots. It's really hard to get it all to unravel and lay out straight. So topoisomerase for the DNA, it can twist and rotate to alleviate that strain that might build up outside of that replication bubble. And so once the double helix parental strands, like shown on the right side, are untwisted and unzipped, now the DNA replication process can begin. We're going to start with what happens on the leading strand. And if you recall, the leading strand is the one that's going to be synthesized in the direction of the replication fork and continuously. So that's going to be shown here at the bottom where it forked off. That's the leading strand. Okay, so let's take a closer look at who's involved there. It's going to start with a primer made of RNA, and this primer is built by an enzyme called primase that adds the RNA nucleotides and joins them together to synthesize this primer. Now, direction is important here, and if you recall when we talked about the structure of DNA, there's a 5' end and a 3' end for each strand. And here this really comes into play because it's only on the 3' end that the new nucleotides can be added. So primase can only add those RNA primer nucleotides to the 3' end. And if you remember, That's because that's where the hydroxyl group is and dehydration synthesis occurs on the 3' end, not on the 5' end where there's a phosphate group. Okay, so remember that detail. This is why we spent time talking about it in a previous lesson. Once the RNA primer is completed, then another enzyme called DNA polymerase III is able to add the DNA nucleotides. And this is where the DNA replication truly begins, because the DNA nucleotides are going to be the actual make up the DNA that's synthesized here. The RNA will be replaced with DNA nucleotides, but not until a later step. And so we'll come back to that after we look at what happens in the lagging strand. And if you recall, in the lagging strand, the synthesis is in the direction away from the replication fork, and it's not continuous. Instead, fragments of DNA are going to be made, and ultimately they'll stitch together to make the full DNA molecule. So like we saw with the leading strand, the enzyme primase builds the RNA primer, and it continues to do that until the primer is done. And the primer is basically just a sequence of RNA that indicates where DNA synthesis is supposed to begin, and where DNA polymerase III is going to hop on and start adding the DNA nucleotides. So just like we saw with the leading strand, DNA polymerase III is also a player here, and it adds the DNA nucleotides right after the primer. And in the lagging strand, this is going to be done in fragments. Each fragment is called an Okazaki fragment. And there's going to be several different places where this is happening simultaneously. Once the DNA polymerase III reaches the next RNA primer, the next fragment, it's going to fall off, and it's going to go find another place to bind. So here you can see it fell off in the step 3, and it attached in a different place, and it's going to continue doing this until all of the bases have their complements. But it has to stop when it reaches the next RNA primer, because that's not DNA polymerase III. DNA polymerase III's job is not to manipulate the primer. It just knows to stop when it reaches the primer. Now, when that happens, DNA polymerase III falls off, but a different enzyme called DNA polymerase I is going to attach to the parent template and the daughter strand that are complementary of each other and bonded together with hydrogen bonds. And so DNA polymerase I is now going to replace the RNA nucleotides from the primer with DNA nucleotides. And like we saw with DNA polymerase III and RNA and the primer made of RNA, these can only be added on the 3' end. That is always the rule here. These enzymes can only act on the 3' end. Okay, so once the DNA nucleotides have all been added and there's no more RNA, there are these little gaps in the backbone, the sugar phosphate backbone, that have to be repaired before the daughter strand is considered completed. So there's another enzyme called DNA ligase that covalently bonds the sugar phosphate backbone between each of the Okazaki fragments. And once that process has finished, then DNA replication of the lagging strand is now complete. Although DNA replication is completed, sometimes there are mistakes that are made during the copying process of DNA. Sometimes the organism's own mechanisms can detect and repair those mistakes, but sometimes they don't get corrected, and it can lead to a mutation. Maybe that will be a benefit, but usually it's not. So in any case, there's another group of enzymes that are involved in correcting errors that might happen in the DNA synthesis process. So one enzyme involved is called nuclease. Nuclease can remove damaged sections of DNA like little scissors. It just kind of goes in there and snips out the damaged section. And so the example shown here is something that happens when there's too much ultraviolet radiation exposure that can distort DNA, causing what's called a thymine dimer. It's where you have these two thymines and adenines next to each other, and it forms like a kink in the daughter strand. So the nuclease can detect that and remove it. But now there's missing nucleotides that need to be replaced. So DNA polymerase comes in and replaces the missing nucleotides. As we saw before, it's going to do that on the 3' end, always. That's where the new nucleotides are added. And it will not miss any. So like the one that's shown here, there'll be one put there also until each of the bases on the parental strand has a complement on the daughter strand. But like we saw with the lagging strand, when you had those fragments, the Okazaki fragments, they needed to be stitched together. So DNA ligase, which we saw before, is the enzyme that can repair the sugar phosphate backbone through the dehydration synthesis reaction. And again, that would only be on the 3' end where the hydroxyl group is found. So sometimes mistakes are made. It's not a 100% perfect process. The error rate after this proofreading process is pretty low, but it's not zero. Sometimes, as I had mentioned before, there could be an error in the copying process that leads to a mutation. And many times mutations are not beneficial, but in some cases they are, and they could actually be the source of genetic variation that helps an organism to survive. As environments change, some organisms have a better ability to survive, let's say like warmer climates, and they're the ones that are going to pass on their DNA to their offspring. And so over time, sometimes these mutations can actually change populations and lead to new species, even if there's enough change over time. So it's not always bad, but usually it is important that these errors get caught and corrected.